My name is Sarah Bowles, and today is March 27th, 2024.
One specific example of a prokaryotic-eukaryotic interaction is the relationship between the bacteria Escherichia coli, more commonly known as E. coli, and human intestinal epithelial cells. E. coli is a gram-negative, rod-shaped bacterium that colonizes in mucous membranes of the gut, intestine, and urinary tract of humans. While many strains of E. coli are harmless and even beneficial, certain strains possess distinct virulence mechanisms and virulence factors that are used to disrupt the host intestinal epithelial cells that can cause diarrhea and other intestinal inflammation. When pathogenic strains of E. coli come into contact with the epithelial cells lining the intestines, they utilize various mechanisms to adhere to and invade these eukaryotic cells. E. coli pathotypes can attach to the host intestinal epithelial cells using fimbrial attachments such as flagella and pili, or afimbrial attachments using surface-binding proteins. These surface-binding proteins, known as adhesions, recognize and bind to specific receptors on the surface of the intestinal epithelial cells. Once adhered to the host epithelial cells, the E. coli pathogenic strains start pathogenesis by producing virulence molecules. This may involve the injection of bacterial proteins, such as toxins, into the host cell through specialized secretion systems. These injected proteins can disrupt cellular processes, leading to changes in the host cell's behavior and facilitating bacterial colonization and survival. E. coli may also exploit host cell receptor-mediated endocytosis mechanisms to gain entry into the cell. When E. coli is able to invade the host cell successfully, some strains have the ability to form microcolonies or biofilms on the surface of intestinal epithelial cells. Biofilm formation allows for E. coli to access nutrients for growth and replication inside of the host cell. The bacteria is able to avoid the host immune system and thrive in this environment. Upon invasion of E. coli into the intestinal epithelial cells, the epithelial cells may activate certain defense mechanisms to limit the effects of the bacteria. These defense mechanisms may include utilizing immune signaling pathways and producing antimicrobial peptides. The host may attempt to alter its structure via tight junctions to prevent further expansion of the bacterial colonies and to keep its protective barrier intact as much as possible. The interaction between E. coli and intestinal epithelial cells is dynamic and can have significant consequences for both the bacteria and the host. For the bacteria, successful adherence and invasion allow for colonization of the host intestine and the potential for disease progression. For the host, E. coli infection could lead to symptoms ranging from mild gastroenteritis to severe diarrheal illness, and in some cases, life-threatening complications such as hemolytic uremic syndrome. Some E. coli strains obtain virulence factors through DNA horizontal transfer of plasmids, pathogenicity islands, transposones, and bacteriophages that can cause intestinal diseases. The E. coli pathotypes were originated from environmental reservoirs, such as from irrigation of contaminated water, and can cause illness such as diarrhea. Once these pathotypes from the environmental reservoirs and some E. coli strains obtain virulence factors, they are able to adapt to new niches in the intestine. Understanding the mechanism for the interaction between E. coli and intestinal epithelial cells is crucial for developing strategies to prevent and treat E. coli-related infections. This knowledge can inform the development of vaccines, antimicrobial agents, and other interventions aimed at mitigating the impact of E. coli pathogens on human health.